All right. Today I'm going to teach you about Ericoid mycorrhizae. Do you know what those are?
No, never heard of it, especially not 5 minutes ago from the first take.
Yeah, and probably from me never shutting up about my huckleberry experiment. So,
Even still, I'm still not sure exactly what they do.
yeah. So, mycorrhizae, the words breaking down into fungus and roots.
I guess you could
Root
say
word.
breaking them down to the root word.
Haha. Are the fungal microorganisms that live in soil in symbiosis with plants. Many plants could not survive without them.
Really?
Yeah. And so the notable important types of mycorrhiza are arbuscular mycorrhiza, which I think are the most common and attach the widest variety of plants. Ericoid mycorrhiza, which I will explain momentarily. Orchid mycorrhiza, which as far as I know, just go with orchids because orchids are ancient and weird. And monotropoid mycorrhiza, which also go with some orchids, which makes things especially clear. So, the important thing about mycorrhiza is that they allow plants to take up nutrients because otherwise they would not be able to. Like they can do it, but like poorly. And it's not all plants, but like a lot of plants. So they break down organic material and allow plants to take up nitrogen and amino acids with their root systems.
So they just like live on the roots
Pretty
and
much.
just
In, on, around, it's all there.
and a bunch of plants just wouldn't be able to even do anything.
Yep, they would just die.
That's crazy.
Yeah, it really is. And so the way that they work is that they invade root cells and kind of just live inside the plant roots. And then they slowly branch out their little hyphae networks and then profit. So they are like able to like decompose organic material that the plants wouldn't otherwise be able to access nutrients from and whatnot. And in return, the plants offer the mycorrhizae basically food via sugar. Yeah. So yeah, it's really interesting. Ericoid mycorrhizae, which are the ones that I'm specifically talking about in this presentation, which are a very diverse array of mycorrhizae. They specifically live in symbiosis with the Ericoceae, yeah, something like that. Anyway, so the plants of that family are understory plants primarily, and also like heath plants, like heathers. And they're actually responsible for 20% of the carbon storage that plants are contributing to on Earth currently. So they're pretty important.
Whack.
Yeah. And some
get rid of all of them for no reason. That would be smart.
Yeah, that would be smart. So the notable members of the Ericoceae, which is what I'm going with. I'm a Latin student. You're a Latin student, and I don't think either of us know how to pronounce that. The rhododendrons, heathers, and other heath plants, and then like blueberries, cranberries, huckleberries, and a bunch of other like forest berries. And so in experiments, Ericoid mycorrhizae have like a lot of these plants don't grow from seed very well. So like if you take cuttings of them, mycorrhizae also help them to regrow roots, which are called adventitious roots. Yeah.
That's
which is
also kind of crazy that
yeah.
just help them grow their own roots.
Yeah. And like their Ericoid mycorrhizae are really, really specific to this family of plants, and all of these plants need them to basically survive and reproduce properly. But they can also interact with other, like the Ericoid mycorrhizae aren't as necessarily specific to the plants. They can actually interact with other plants that end up kind of in this area with the Ericoid plants. They'll just kind of opportunistically be like, hey, there's a new plant here, I'll hang out with this one as well.
Just use this plant for,
Yeah.
I'll give it nutrition and it'll give me sugar in return.
Yes, exactly. And the really interesting thing about this coevolution, which has taken place since essentially at least the beginning of the Ericoid evolutionary branch, is that these Ericoid mycorrhizae actually have a smaller genome because like the plants compensate for some of their needs. Yeah.
So it's kind of like mitochondria.
Yeah. Yeah, exactly. This like symbiotic relationship, in some cases like endosymbiotic, because they live inside plant cells for at least the first stage of their development. Yeah, so they're basically, plants compensate for part of their like lacking genome, and like, so basically they're obligate symbionts. They can't really live by themselves, but they can live with a wide variety of plants. Because all of this plant family can't survive without Ericoid mycorrhizae, it's like a very important part of the ecosystem. So like one example that I was reading about in one of the articles was in the South Africa heathlands, which like basically the vast majority of plants in that landscape rely on these Ericoid mycorrhizae to survive. And like if something were to damage that relationship, that entire ecosystem will wipe out. Yeah. And the actual way that like the Ericoid mycorrhizae interface function occurs is still not entirely known. There's a lot of research into it. It has not elucidated a lot of things. The Ericoid mycorrhizae compared to other mycorrhizae, like the other mycorrhizae, so there's like different stages of growth. The like intraradical stage, which is when they're inside the root being a radical root. It's a pretty radical word.
A radical?
Yeah.
What's that?
Radical just means root.
Oh.
Yeah.
Oh.
Yeah.
Okay, nothing to do with free radicals.
No.
Which I don't remember what those are.
Actually,
either.
I wonder if it does. We'll have to look that up later. But
those are like electrons, right?
I mean, kind of like oxygen radicals are yeah. But anyway,
off topic.
Yeah. So they have an intraradical stage, which is when they're living inside the plant root and like developing, and then they have a lot of mycorrhizae have a periradical stage where they create their own like layer of stuff around the root. Ericoid mycorrhizae don't do that. They just send out their hyphae, which is the extra radical stage. And like that's like they're not sure why they're missing that middle stage and all that stuff, but the hyphae allow them to like break down and get nutrients from the soil and then bring them into the plant. Yeah.
Putting out their own roots from within the roots.
Yes. So now we get to the not like the important, but like the exciting part of the presentation, which is my research, huckleberries. So huckleberries, super delicious and nutritious. They have huge cultural and economic significance for the entire Northwest and especially Montana. Why have they never grown successfully in captivity? We have tried a lot of things.
Because it would be very profitable if that they could actually be grown in captivity.
Yes.
Just harvested.
Yes, because huckleberry picking is very labor intensive, which is why huckleberries go for like $50 a gallon.
Which I actually only learned about when you started doing this project and you told me about this, and I was like, oh,
yeah,
really?
Yeah.
That doesn't make sense why they're so expensive.
Yeah. So they've never grown successfully in captivity. We've tried things like the effects of um, light, sterilization, cold, like all of these different things, and they just do not grow very well. They never, like, even if they're able to like take hold and start growing, which is like not usually very successful, they almost never fruit in captivity. And so there's this idea that like there's something missing in our laboratory and garden settings that huckleberries need that is in the wild that allows them to grow. And so the idea is that it might be the Ericoid mycorrhiza that they're missing in like a garden setting. And so that's like the potential linchpin for the huckleberry domestication hypothesis. And so like we kind of figured this out because blueberries, I mean, they're of course Ericoid. Blueberries cannot fruit in most conditions unless they have Ericoid mycorrhiza. So, and then when they figured out like, yo, when we did this thing where we dug around these huckleberries, there's Ericoid mycorrhiza here in the wild. Wow. Yeah, that's one of the ideas that it is probably the missing thing.
Somebody should do a project on that.
Yeah. So huckleberries, why huckleberries are important and why should we bother to domesticate them? Well, so they play a very vital role in their habitat. They have a lot of economic and cultural significance to us, and especially Native Americans. And we have a lot to learn from their interspecies symbiosis with mycorrhiza and other animals. I mean, I guess mycorrhiza is not an animal, they're other organisms. So huckleberries have had traditional native use essentially since Native Americans came to America like thousands of years ago. They're both used as a food source and also have medicinal use. They're especially an important food source in the winter months because they're able to be dried and kept through the winter. They're a major part of the summer tourist economy in a lot of the Western US and Canada because they draw in hikers and stuff like that into national parks and other areas, like especially like ski resorts and stuff like that during the summer. And like, I mean, everywhere in Montana, you see huckleberries, like gift shop, all that stuff. Because huckleberries are really unique to like these mountain areas where like they don't grow anywhere else, like which is crazy. And they're being threatened significantly by climate change, which is an issue that we'll get to. Yeah, sad. So they have massive ecosystem value because such a wide variety of animals feed on them. So that's like bears, deer, rodents, birds, insects, like everything is eating huckleberries. And from their nutritious value, we really haven't been able to investigate that enough, even though like, so blueberries are like super good for you and have like a lot of antioxidants. Huckleberries are like blueberries, but even more concentrated. And so like there's a lot of like potential nutrition value in huckleberries that we would not be able to access with like they're not readily available to us to like sell and use. And like huckleberry picking is a fun outdoor activity, family bonding, stuff like that. It's like, I mean, in a lot of places they have like huckleberry festivals and stuff like that. It's like a kind of a major tradition in a lot of these areas, and like the massive droughts and like extreme temperatures that we've been having have been like decimating the huckleberry harvest, which sad and not good for animals and not good for people. So like I mentioned before, they're a super important summer food source for a lot of different animals, but especially in the way that they allow them to like build up their fat stores to survive the winter.
Mhm.
Like, since especially huckleberries grow in super mountainous areas, they are in areas where the animals will have to like hunker down for winter, and the huckleberries
Mhm.
survive the winter too. Um, but
Huh.
like, so cold is nothing basically to huckleberries. But yeah, without that ability for those animals to build up their stores of fat and stuff like that for hibernation, or for birds just like day-to-day energy, since birds have such fast metabolisms and rodents do, like it,
Hmm.
that they would probably start to have a harder time surviving the winter.
Mhm.
Huckleberries are also very interestingly a burn recolonizer. So like huckleberries grow really well in burn clearings, which is why Native Americans
Oh.
used to actually clear parts of the forest, like little clearings, with burns to allow huckleberries to grow better.
Huh.
So yeah, and they were used for Native peoples throughout the year. Fascinatingly, huckleberries were one of the first plants to recolonize the Mount St. Helens eruption areas, which
Oh.
are really interesting because like that's complete wipeout of habitat. But like it's believed because huckleberries have such an intense like widespread root network, that they were able to just kind of, especially in burns and stuff like that, they're able to just grow up from below.
Just kind of like
Yeah,
clone.
yeah. And so like and also with the mycorrhiza and stuff like that, they kind of provide like a really important micro ecosystem for a lot
Mhm.
of different species, which is really cool.
So when the huckleberries recolonize a place, they're
Mhm.
also bringing that mycorrhizae with them?
Yes, yeah, because the mycorrhiza in theory survive with those root systems.
I see.
Yeah.
And spread with the root systems when they spread out.
Yep.
Huh.
Yeah. So yeah, one of the struggles is like people have had more success growing huckleberries in captivity from cuttings than from seeds, but they never fruit. And like,
Mhm.
because from seed, like without that mycorrhiza like supporting their development, it's like really hard for them to like really grow and survive
Mhm.
and thrive long term. So like cuttings have had a little better success, but like not much. And theoretically, it's because of that Ericoid mycorrhizae, which is specific to the huckleberry plants, is
Yeah.
like missing. So yeah. And so this is my project. So my hypothesis was that we might be able to grow huckleberries in a lab setting if we take native soil from around huckleberry plants. Like in my case, I basically drove to Idaho and went to where I recognized a bunch of huckleberry bushes and gently dug up soil from like around them and around the root system, and then like dumped it all in a bucket. I think I had like 3 5-gallon buckets of dirt that I collected from all around the area where I was collecting. And so like the idea is like how to incorporate the Ericoid mycorrhizae into the soil, especially when they're species specific to the huckleberry.
Mhm.
And like the only way, in my opinion, to do that was to not incorporate it into the soil, but take the soil that already has that mycorrhizae in it theoretically,
Mhm.
and hope for the best. So my hypothesis was I thought that huckleberries may. So I wanted to do growth, but it ended up just focusing on germination because we didn't get germination until like a month and a week in, which was like kind of a challenge because like in other circumstances, because it's usually not germination that's the problem, like that germination usually happens within 2 weeks, and for dust it took a little longer than that. So the growth we'll have to monitor more long term. But the idea was like, we can monitor the germination and growth and see if it's better in the native soil environment than just like a typical peat pellet, which is what I ended up using, Jiffy 7 seed pellets, and versus the same Jiffy 7 seed pellets, but with just like a generic mycorrhizal supplement, which I dissolved in water. I guess the biggest challenge ended up being that it germinated, like the seeds actually started germinating pretty late game, and so later than I expected. And so we don't really have a chance, at least within this semester, to get statistical significance on that, because we're getting more and more germinating every day. And like, so like for this semester and for my plant research class, it's kind of too late to get like the real statistical significance, but at least we have a couple. I think it's crazy that they germinated at all, which like,
especially after like a month of waiting.
Just checking hopelessly every day, watering like these pellets, and like it's like, ooh, some green. That's moss. That's algae.
I think that was like the first day I came in. I think that was like, I the one who was like, wait, are those sprouts?
Yeah.
And then you were like, wait a minute, those are.
Yeah.
Actually, I do have a question about getting soil. So the mycorrhiza, you said they live in the roots, right?
Yes.
Do they also like spread into the soil?
Yeah, with their hyphae, at least they do. And like theoretically, since they're a fungus and since they have to like invade and colonize the cells in the first place, like presumably there's at least some, I guess, spores like hanging out in the soil. Though I'm not entirely sure on that, and then again, neither of the scientists. So
what better way than to try it?
Yeah, exactly. Because I didn't want to like actually damage the wild huckleberry roots and stuff, because that would be, I think, kind of unethical of me, and also like I was on not my property to do that, so we're not going to go into that. But I don't think they're going to be missing a couple gallons of dirt off of resort land. Anyway, so in my methods, I had 3 separate planting conditions, which was the native soil, the standard Jiffy pellets, and the Jiffy pellets with the additive. I still use the pellets for the native soil, but I just dug all the peat moss out and put the native soil in. And I planted 192 huckleberry seeds, which actually wasn't even like, there's so many seeds in huckleberries. So I had to go through huckleberries and just scrape all the seeds out. And like, there are like at least a couple dozen seeds in every huckleberry. So like, I think I probably extracted like 500 some, maybe if not like 1,000 seeds from all the huckleberries that I dug through, and then figured out like I'm only going to be able to plant like 4 seeds per pellet, because they're only about like 1.5 inches like in diameter. So like putting any more in there would be like not only hard to track where they are, but like so I only ended up planting 4 seeds in each pellet.
Actually, I have another question.
Yeah.
So do huckleberries like reproduce and spread through like the seeds and the berries, or is it primarily like root system cloning?
As far as I know, it's both.
Okay.
So like the one thing about root system and cloning is like in those areas where you're competing for resources like water and nutrients and sunlight, you don't want to be competing with ones that are genetically similar to you. And so like because otherwise, like, it's not evolutionarily practical, because you want like your descendants to survive, so you don't want to compete against your descendants.
Right.
So that kind of comes in with animals and animal digestion, especially. So there was this hypothesis that possibly huckleberries needed to be digested by an animal like digestive system in order to sprout, similar to like some other plants. And they found that it actually doesn't make a difference whether they're digested or not, but it does make a difference that the fruit from around it is removed. So like the benefits of animal digestion, instead of being like the digestion process being necessary, is removing the fruit layer and spreading them out and away from their relatives. Yeah, so like the bear eats it.
Very interesting.
In one area, goes somewhere else, poops,
Yeah.
and then the huckleberry won't, that can sprout from that, will not be competing against its own like family members essentially to survive. Yeah. And so like
so
I
planted the
seeds. I checked them daily for like, I guess it's been almost 3 months now, and monitored like who's germinating, mostly who's germinating. The growth's pretty insignificant at this point. They're barely like, they're not even like half a centimeter tall. They're super, super tiny, the ones that have germinated so far. And so like we just kind of had to abandon the growth angle entirely and just focus to see if the soil condition affects germination, which like at least gives us the information whether or not it affects germination. And then we did all the statistics in our studio, which you helped me with because I couldn't remember half of statistics class.
And I couldn't remember a quarter.
And so as of the results of that time when we did it, we had 8 of the standard control seeds germinated out of the 64, 9 of the native, and 3 of the mycorrhizal generic supplement seeds. And so the total germinated across the conditions on, I think it was the 9th of December, was 20 out of the 192. And so like the statistics we did were like pretty off, or like I guess just like based on what we had. And like there's, since we're getting more germinating by the day, it's hard to say, like we'll have to watch in the long term to see like if there is actual statistical significance or not. And like we were also measuring whether there was like a difference in the time to germinate, but we found no statistical significance in that either with our little graphs here. So
like significant at the 20%.
Yeah, I mean
20%. I like
in biology we tend to aim for the 0.05, but we got like honestly at the 20% like not bad.
It's good for like this sort of experiment,
Yeah,
but mostly it's indicating that we may have something here. We should keep watching.
Yeah, and like Dr. Almquist said, even if we find out that there is no statistical significance between the 3 types of growing conditions, then we at least know for that, like for the germination part, soil doesn't matter. You can germinate in anything, and then be able to maybe transfer to native soil if we find out in the long run that native soil like actually helps huckleberry growth.
That would be interesting.
Yeah, so we do see an interesting potential thing here with the mycorrhizae supplement being like possibly, I mean, we don't know for sure until we have more, but for the mycorrhizae supplement to maybe be detrimental to the germination, which would be interesting. And I would guess that if that is actually the case in the long run, that it's possibly because the mycorrhizae aren't species specific to the huckleberries. And so like generic mycorrhizae, not gonna help, maybe even harm. So yeah. And they all have been germinating in the same period with like new ones in each one every day. So like the timeline's pretty much the same. We haven't seen any significance there either.
Oh, what is the y-axis there actually?
Oh, I think it was This was days.
Okay, and
Yeah, they all didn't go, like, we got our first one around, like, I don't know, like 26 days or something, and then didn't get any for like a couple days. Then, like, yeah, so fun times. Um, yeah, we've our huckle babies, they're very small. They only have, most of them only have 2 leaves, but as of the other day, some of them now have 3 leaves. And like,
Ooh.
it's kind of hard to tell the scale here, but like this entire thing is only about like 1.5 inches. So these are tiny, tiny
Yeah.
sprouts. But we will continue to watch them, see if we get anything interesting. Unfortunately, I wish we could have like maybe cultured the soil to just see if we could find those Ericoid mycorrhiza, but fungi are like weird to culture in a lab setting. Like, we weren't
Mhm.
sure if it was contaminated, so it was like, well, we'll just stick with what we have. So,
Yeah.
but I guess if I could go back, that's one thing I might try to do. Um, yeah. So now we have 11 standard, 11 native, and 7 mycorrhiza, and I believe that was as of yesterday. So I will have to go back and check again today and see if we have any new ones. But
So it looks like the supplement is maybe catching up, but
it might be. We have no idea. And once again, we have like 192, and that's like 29
oh, wait a
of
minute. No,
those.
wait a minute. That's like the standard has 3 new ones. The native has
Yes, 3 new
2
ones.
new ones, and the mycorrhiza has 1 more.
Has the mycorrhiza does have 3 new ones, at least since the last time. Yeah. So that's something.
Might be something.
It might be something. Yeah. So yeah.
Very interesting.
These are my references. We had the this was just the generic huckleberry domestication and things we have tried. And like, what do Ericoid mycorrhiza do in huckleberry root systems? And then this is where I mostly learned about the genome and the structure and function, which was they I guess they got the structure. The function was a little more, yeah. Um, then like more about the ecology and the host specificity of Ericoid mycorrhiza and how they were spreading out into other plants in the same ecosystems, and like their symbiosis and how it might have happened and how it helps cutting growth, which is interesting. So yeah, that is my presentation. Do you have any questions?
I think I asked most of my questions throughout the presentation.
Cool.
Very interesting, very cool. Hope we get some stuff out of this.
Yeah, it'd be pretty sick to be like, have maybe found a solution to the domestication thing.
Yeah.
And like the cool thing about maybe being able to grow them in captivity is like we can study them more and figure out what they need in their environment to like stop them from dying of climate change. So because it's not good when things die of climate change.
Yeah. It's not good when things die of climate change.
Like humans potentially in the long term. We'll see. So yeah.
We can only hope.
We can only hope.
I'm sure the nematodes will be fine.
I'm sure the nematodes will be fine. The nematodes will make it.
All right.